So, and please ignore the hump on my back. No pockets, that's the downfall of women's clothing. Okay, well, this has certainly been a very interesting year, hasn't it? We've seen a CBD drug make it through the US FDA. How many of us thought that would ever happen? GW Pharma, Epidiolex, and it's for epilepsy. And that's certainly changed the horizon for all of us. So CBD is now in kind of a gray zone. We know that if you go through the FDA rigorous steps that you would be able to get approval, following in the trail blazed by GW Pharma. We have a pending 2018 Farm Bill, so we don't really know how CBD and hemp will be dealt with in that, even though we're assured it's going to be happening. We have the World Health Organization reviewing international drug laws right now that have, this very month, and specifically for cannabis, CBD, and THC, that's kept this plant and these rather remarkable molecules locked away in a box and in the black market, basically. So these are some of the things I'm going to be talking about today. I should also mention, besides not knowing what the Farm Bill is going to do, now that Jeff Sessions is out, we also don't know who the new Attorney General will be. So there are a lot of changes in the future. So first we're going to talk about some basics, some chemistry. We'll talk about the domestication syndrome that's been going on for the last 50 or 70 years, the need for expanded chemobar classification. I'm going to touch on phytotherapeutics, just because I'm so incredibly interested, and then a little bit about analytics. So from the basics, so this is chemistry, right? So we have been on a rather amazing trajectory. So in the 70s, we had the characterization of the first two plant cannabinoids, phytocannabinoids. So phyto means plant, and that's CBD and THC. So these are secondary metabolites. So cannabis makes lots of secondary metabolites. These happen to be very abundant, and just like how we've transformed the milk cow that used to make a gallon of milk, now milk cows make pounds, 80 pounds of milk. So now we have cannabis plant that used to make 3 or 4% of THC that is making upwards of high 20s. And here in Vegas, where there's a little magic, we actually see cannabis flower over 30%. So it's really extraordinary how this plant has been manipulated. Now, you probably know that these secondary metabolites, and I should have put a photo in here, are produced in the trichomes of the plant, so these glandular structures that are on the mature inflorescence of the plant. And that's actually where terpenoids are also made. Well, so here's the parallel I want to draw for you, and you've probably been exposed to this to some degree, but your body also makes cannabinoids, and those are called endocannabinoids. And they have mirror images of THC and CBD found in the plant, and no surprise, that's why cannabinoids from the plant can have such a major impact on our bodies, both physiologically and pathologically. So the two compounds that we make are 2-AG and anandamide. 2-AG mimics the role of CBD. It's a ligand for the CB2 receptor, which is present more peripherally. That means it's not in your brain, it's out in your lymph system, it's in your immune system, it's in your blood, it's in lots of tissues, and anandamide, which is similar to THC. And that word actually means bliss. And it is pharmacologically similar to THC. on receptors, but it also interacts with a lot of other receptors, as I'm going to point out. So the other large class of chemicals, so that was the two major cannabinoids, the ones the plant makes, the one your body makes. Same thing is going on with terpenes. So this is something that you may not know. It's really rather curious. So through evolution, as Mother Nature found molecular combinations that were successful, they were kept through evolution into other species. So terpenes are actually what give the plant its aroma, and it's what is distinguishing. Cannabinoids have no odor, and as you all know, cannabis has a lot of odor and a lot of distinctive odors. And those distinctions come from the unique combinations, or maybe not so unique, combinations of terpenes that are present in a particular strain or cultivar. So just like the concentration of cannabinoids has been going up, the concentration of terpenes are going up. So originally about 1% of a plant would be terpenes, now we see up to 3.5%. And there's also this phenomenon going on where producers will be adding back terpenes at abnormally high levels. But what I have to say is aromatherapy is very real. These chemicals are perceived in your olfactory sensors, and the receptors there, and those receptors are throughout your body. So these terpenes that you take in through ambient air will reach a physiological level that you can measure in your blood. So they're moving around your body and they're doing things. So I said the plants make terpenes, your body makes terpenes. Here are terpenes made by your body. So cholesterol is the building block for the sex hormones, testosterone and progesterone. Cholesterol's been getting a really bad rap for a long time, and I'm really sick of it because it's an incredibly essential molecule that your body makes, not only to be the precursor for these two important chemicals, but also for just basic health in your body. All right. So now we're moving on. We have a lot to cover. I want to talk to you about the domestication syndrome. This is a word that was coined by Paula Brown, who's a professor at University of British Columbia in Kelowna. And it's used to describe this long history of informal breeding and cultivation that's been going on with the cannabis plant. And amateur breeders were selecting for one thing, which was high THC. So the consequence of that is a loss in chemical diversity and a loss in genetic variability in the plant. So when people talk about strains, it doesn't really mean anything anymore. Strain is more for branding. And we should think of strain naming as branding until we create a set of criteria that have to be met for you to call this plant a blue dream or an OG Kush or whatever. And it's really important, just like Ken was saying, the industry is evolving rapidly, and we have to adhere to certain standards so that there's more credibility. But clearly, classifying cannabis strains simply based on the ratio of THC to CBD, which is largely done now. High THC is a drug type 1. There's drug type 2, which is about equal levels THC, CBD, and type 3 is high CBD. Clearly, we need to get a little more sophisticated. We actually did a large large study here in Nevada. We took 5,000 individual flower samples that represented 403 different strains by name only. We found that they were 98.3% identical by cannabinoid content. So you can see here, we were looking at 11 cannabinoids. So what I have to qualify here, and for analytical science, you only are going to find what you're looking for, right? So there are 12 certified reference standards for cannabinoids, so you can look for 12. You can only quantitate 12. You can look for more, but you can't quantitate them because you don't have the reference. So in this study, you can see all very high THCA. However, when we looked at the terpene profile, and this is 22 different terpenes, you can see these 403 strains, 5,000 samples, clustered into 3 unique groups. So these 3 terpene clusters have very unique terpene profiles. And while this industry is operating under this arbitrary, geographically restricted regime, which is just nuts, we don't know, but we highly suspect, because we're not the only ones doing terpene chemoprofiling, that these same types of clusters are existing out there. Mark Lewis and Eve Russo have also done terpene cluster analysis, and I think they feel there are also 3 to 4. They didn't see what they showed it. So what is the solution? So as I said just a moment ago, I think strain names should be reserved for branding purposes until we are all as an industry ready to adopt more standardization of agronomic breeding practices where we keep track of parentages, where genotyping is required, and expanded chemotyping. And this is just a couple of quick studies I'm showing you to prove the point. Again, this is from the University of British Columbia. They just looked at 33 strains, and when they expanded the metabolomics, or when they were looking at what falls into the region of cannabinoids and confirming by mass spec that it has the right structure, they saw 21 unique phytocannabinoids that weren't being characterized previously, and only 3 of those were in high THC strains. The rest were all associated with CBD strains. So again, this is because we've been breeding, crossing, hybridizing hybrids with hybrids, all for high THC, and the consequence of that is a loss of genetic variability. And so that variability is more obvious when you look at CBD strains. Now, here's even a larger study. There's a very active academic group at the Technion Institute in Israel that have characterized actually 105 different cannabinoids. In this study, they looked at 94. So 94 is right, it's not just THC and CBD. And they used these to profile 36 of the strains that are allowed to be grown in Israel for their national medical marijuana program. And what they went on to show is if they took genetically identical strains, grew them in 4 different greenhouses, same strain, 4 different locations, that those plants produce different cannabinoid profiles. So there's a concept in nature or in science called nurture versus nature. So you have the genetics and you end up with a phenotype, but the influence of the environment can impact what you actually end up with. But this also brings up another complication of genetics now. It's much more than just the actual sequence of the DNA. The DNA can be modified through methylation. And putting methyl groups on the backbone of DNA changes the genes that get expressed. And these epigenetic changes can get carried along, especially since people are clonally propagating. We don't even know how far these epigenetic changes get carried along, and nobody's really, I'm sure people are looking at it. I just haven't seen anything published. But more importantly, this strain that was grown in 4 different locations, extracted, it was used as medicine, as an anticonvulsant, and they showed that the minor phytocannabinoids, these small amounts, influence the ability of CBD to serve as an anticonvulsant. So it's not irrelevant, especially if you believe that this plant is medicine. So this is simply to bring the point home and for me to reiterate. So there's 2 little peaks of ice up there. That's THC and CBD. And underneath is all of this other capability that's inherent in this plant, and no doubt our body as well. So this is a very exciting time. This is an era of discovery. So I think by next year, we'll all be amazingly surprised of what will be discovered in terms of what's under the water. All right, so obviously, I hope I've convinced you that there's a need for expanded chemotyping. We need to look for more than what we already know. So this is really the realm of science and why this is so exciting to be a scientist now in this industry. We don't even know how these other secondary metabolites will necessarily be interacting from a pharmacological point, nor do we know how it impacts crop diversity or the health of the crop, the ability of the crop to resist pests, or to thrive in different environments. Right now we're practicing big time monoculture, so the risk of introducing a pathogen that can totally wipe out your crop is getting larger. The emergence of new diseases of cannabis is a possibility. So we need more research, we need more analytical in-depth studies to not only identify these new metabolites, but also to figure out how they're interacting with this very complex system of the cannabis plant and our body. So any particular cannabis strain or cultivar, it's not just making THC and CBD, it's making all these chemicals, minor cannabinoids, and all these terpenes, right, the aromatic compounds. And all of these chemicals together, whether you're smoking it or eating it, rubbing it on your body, they're interacting with about 30 to 50 different receptors in your body. This is just a diagram to show how this can be so impactful. So you have receptors on one neuron interacting, so that's presynaptically and postsynaptically, and these molecules go from one neuron to the other. So this entire system, the endocannabinoid system in your body, its main role is to modulate dopamine. So dopamine is the main neurotransmitter. It's how one neuron gets a signal from the adjacent neuron that something's happening. And you have to realize how vastly influential the endocannabinoid system is. I mean, it controls your sleep, your mood, your how you learn, your fertility, what you eat. I mean, it's like the universal control in your body. And the two prime messengers in your body were those two molecules you saw at the beginning, those two endocannabinoids, 2-AG and anandamide. Like I said, there's about 30 to 50 receptors, and they really are controlling the homeostasis of everything that's going on in your body. So what you have to realize is these molecules are being made in a very localized, need-dependent basis within your body. So they're not made all the time. It's only if you experience pain, or there's some stimuli, or you're sick, or you need an inflammatory response. These molecules get made at the membrane. They get dispatched to communicate a message throughout your nervous system or your immune system, and then they're immediately degraded. When you're using cannabis, you're like bathing all of your receptors in abnormally high doses, very high doses, and we have no idea what the consequence of that might be. We know some of them, like I had mentioned that the main role of your endocannabinoid system is to regulate your dopamine response. Well, dopamine is involved in many pathological diseases, Parkinson's being the best. My father has Parkinson's. So that's been when your dopamine system is not firing accurately. You're not making enough dopamine. The dopamine's not transmitting that response through your central nervous system, and you can't move. You can't pick your foot up. It's something you take for granted all the time. That's a dysfunction of your dopamine system from your ECS. And another consequence is drug addiction. Drug addiction is directly tied to dopamine. Dopamine, you can train a rat to push a lever to get a little bit of cocaine. And they do that because that's a reward. So the dopamine system is also a reward system. And that rat will just keep pushing that lever to keep getting that little reward. So these things are all connected in your body. And this is rather a remarkable diagram that a systems biology group actually tried to come up with a computational model of your endocannabinoid system. And I know you can't read it, you don't need to read it, I can't even read it, but it's just to show you all of the interconnections. So the two red ovals at the top would represent anandamide and 2-AG, your two internal endocannabinoids. But remember that these the phytocannabinoids, THC and CBD, interact in the same way. So just imagine, this is actually called the scale-free network. It represents what's called a small world. So it's very robust, highly interrelatable. If there's a perturbation anywhere, the whole system will collapse. So this is something to keep in mind. So let's talk a little bit about phytotherapeutics. So this is the use of plants in therapy. And it's really remarkable that we know as much as we do because of all the federal prohibitions on being able to conduct clinical trials. A lot of us have to experiment on our own and determine based on what either our recreational or pathological needs are, how much THC, how much CBD, and of course none of you are thinking about what about the terpenes, what about a more expanded chemo profile. Clearly, the higher level of purification you go, I think you get farther away from a beneficial I specifically wanted to point out the neuropathic pain and the ARIs, which are maltose reductase inhibitors. And why are those important? Because those are both associated with diabetes, one of the biggest chronic diseases in our society now. And there's been some bad press lately about the use of cannabis if you are a diabetic, and there's been some warnings, and I'm not sure where that's coming from, but CBD specifically inhibits aldose reductase. And why that is important is if you overconsume glucose, sugar, if you're taking in too much sugar, your normal way of dealing with that sugar to convert it into energy gets overwhelmed, and this other pathway takes over. It's called the polyol pathway, and it converts sugar to fructose. And fructose is toxic. So the fructose is in your liver, and so your body wants to do something with it, so they make it into fat. So you get fatty liver syndrome. But it also gets out your bloodstream because the liver gets overwhelmed, so it leads to constriction of your bloodstream, heart disease, and these enzymes also are concentrated in the eye, the kidney, and the myelin sheath, which is what protects your nerves. So that's how you end up with neuropathic pain if you're diabetic, and fatty liver disease, insulin resistance. So I see this as a huge market for cannabis is in the treatment of type 2 diabetes. So there are side effects from using marijuana. I don't know, I mean, obviously you're all here because you're interested in cannabis, and it does cause short-term memory loss, and we know why. We know because it increases acetylcholinesterase, which is in your hippocampus, that's responsible for laying down short-term memory. Good news there is there are terpenes that actually counteract that. Obviously, it affects food intake, cognition, motor response, psychosis, dependence, and tinnitus. I don't know if any of you suffer from tinnitus, but I do. Ringing in the ears, that's exacerbated by cannabis use. So as you see this whole list, if you remember, this goes back to all of that umbrella of the physiological aspects from your own endocannabinoid system. So you can see why all of this is affected by cannabis use, because you're screwing around with that homeostasis. And sometimes it, of course, can be very beneficial, as we just talked about. So terpenes do mitigate some of these side effects. In particular, alpha-pinene is known to lessen short-term memory loss, and we know why, because it turns off that acetylcholinesterase. Alpha-humulene, main terpene in beer, it actually reduces food intake. Alpha-pinene, again, is good for cognition. Psychosis, I know Ken was mentioning psychosis. Well, psychosis is a real thing. There's a certain percentage of the population that harbors certain gene variants that make you more predisposed to psychosis. Again, terpenes are wonderful because they can help mitigate that, because they're very sedative with limonene leading the list. And no help yet with tinnitus. This is not my slide. I took it from the professor of pot. It's a great slide. You know, so often, whoever is responsible for this website, they put out an amazing consolidation of lots of information. So here we have a list of very popular terpenes and the in vivo activities that they affect. and also by what mechanism. I want to emphasize that so much of what we know about specific cannabinoids and terpenes comes from animal studies because of this ongoing restriction with being able to do actual clinical trials. But just like these chemicals, the pathways to synthesize these secondary metabolites exist in plants, insects, us, so do terpenes, and so we see these rather remarkable binding to these receptors that are all involved in sedation. So terpenes overall as a group are very sedative, they're very calming. There are cultures around the world where people use terpenes to actually calm their nerves, to calm down, to relax, and you can see why. And the major neuromodulators in this case are GABA and glutamate. So you can think of these again as neurostimulators, the neuroexcitability. So glutamate is a green light and GABA is a red light. So terpenes, it's pretty remarkable, right, that these plants, not only do we have this massive ECS, endocannabinoid system in your body that the cannabinoids interact with, we have these sedation receptors that the terpenes interact with. I mean, we've clearly evolved with this plant, and we've been separated from it too long. It has amazing benefits to offer. One of the other very interesting things about terpenes is their permeation enhancers. So what does that mean? Well, on your skin you have all kinds of layers